Good morning, students. Today we will further study about the characteristics of gymnosperm. In case of heterosporous condition, where there is mega and microsporangia occur on mega and microsporophyll, which usually aggregates to form a compact cone or strobili. These cones are usually monosporangiate, that is unisexual, but in some species, example in case of Ephedra foliata and Ephedra intermedia, the bisporangiate cone also occur. Male cone are usually short-lived. Microsporangia developed on the abaxial side of the microsporophyll. The number of sporangia per sporophyll varies from 2 in case of Pinus to many in case of Cycas. The development of microsporangia is eusporangiate. What do you mean by eusporangiate? Eusporangiate it means that it develops from more than one initial. The female cones are formed by the aggregations of megasporophyll that is persistent on the plant for several years. The megasporophyll may be similar to normal foliage leaves, example in Cycas, or are colline in case of Pinus. The megasporangium ovule is naked structured on the megasporophyll. The ovule are orthotropous and unitegmic but are bitegmic in details. The ovular integument is differentiated into three layers: outer, middle, and inner layer. The outer and inner layer are fleshy while the middle layer is stony. The microspores are liberated in the various stage of development of the male gametophyte. For example, they are liberated at the 3-cell stage in Cycas, 4-cell stage in Pinus, 5-cell stage in Ephedra. The male gametes are non-motile with the exception of Cycas and Ginkgo. The number of archegonia in female gametophyte is variable. There are several archegonia in Cycas and only 2 in Pinus. The archegonium of nitum is represented by only ovum. The archegonium has a single egg and a venter canal cell. Neck canal cell are however absent. Pollination is direct, that is, pollen grain come in direct contact with the ovule. Pollens are deposited in the pollen chamber where they germinate. All gymnosperms are wind pollinated. Fertilization is siphonogamic and the pollen tube function as a hostoria in case of Cycas or sperm carrier in case of Pinus. The zygote formed as a result of fertilization is the mother cell of the next sporophytic generation. The development of embryo is meroblastic, that is, the embryo develop only from a basal part of the zygote. Development of endosperm take place before fertilization and hence the endosperm is haploid. With a few exceptions, example in neonatum and Welwitschia, there are 4 nuclear divisions in the early stage of embryo development. The embryo is endoscopic, that is, shoot apex is directed opposite to the micropyle. There is a marked tendency of polyembryony and several embryos developed in a female gametophyte. The polyembryony may arise by the fertilization of more than one egg or by the division of the zygote, cleavage polyembryony. But due to physiological competition, only one embryo attains maturity. The naked ovule develop into seed. The ovular integument form the seed coat. The number of cotyledons in a seed is 1 or 2 as in case of Cycas or many in case of Pinus. The seed usually has a long or a short dormant period and then it germinates to form a new sporophytic body. Seed represents 3 phases in the life cycle of gymnosperm. Integument and the nucellus represent the mother sporophytic phase first sporophytic phase, endosperm the gametophytic phase, and the embryo is the next sporophytic phase as it develops from the zygote. There is a distinct alternation of generation. The diploid sporophytic phase is dormant, whereas the haploid gametophytic phase is reduced. The gametophytic phase is dependent on the sporophytic phase. Thank you, children.